In deciphering the genetic code, we have scientists Marshall Nirenberg and Heinrich Matthaei. They performed experiments with E. coli cell-free extract, which is just blended up cells, and they get the cytoplasm and the ribosomes from that. With this, they added poly-U mRNA, which is a bunch of the uracil nucleotides, and they added 20 amino acids. One of these amino acids was radioactively labeled. They allowed for these mixtures to incubate and for protein synthesis to occur. In the end, they examined the results in 20 different tubes. One of these tubes had a radioactive amino acid, and therefore the protein that it synthesized was also radioactive. For the poly-U nucleotides, it was phenylalanine. From this, they discovered that UUU codes for the amino acid phenylalanine. With more experiments, they discovered CCC codes for the amino acid proline, and AAA codes for the amino acid lysine. GGG does not code for anything. Now they wanted to determine the length of a codon. Scientists Marshall Nirenberg and Philip Leder performed an experiment containing two mixtures. One contained poly-U mRNA, amino acyl tRNA bound to radioactive phenylalanine, and ribosomes. The other mixture contained all of this except for the poly-U mRNA. They poured these two mixtures over cellulose nitrate filter paper and performed a wash after. The cellulose nitrate filter paper would catch the ribosomes and whatever was bound to them. From this, they found that the filter paper was radioactive for the mixture that contained the poly-U mRNA, and the filter paper was not radioactive for the mixture that did not contain the poly-U mRNA. They were able to identify 61 codons using this scientific method. They determined with their results that if the filter paper was radioactive at the end, then this meant the trinucleotide that they used coded for the radioactive amino acid that they used. So in the case of this experiment, it meant the trinucleotide poly-U mRNA coded for the radioactive phenylalanine.